Good morning, everyone, and welcome to today's Chaga Echo session. Please know that we are recording the session for today for distribution on the Chagas Echo website, so you might see that prompt going off in just a second, or if not, it already has. We are excited to have everyone join us today for this series. Today's session is brought to you by Rockefeller University, Clinical Directors Network, Texas State University, San Diego State University, and UT Health San Antonio Echo. My name is Maria Gutierrez, and I'm with the San Antonio Echo Super Hub. I do have some announcements before I pass things over to Deliana to continue with some introductions of today's speakers. Okay, so we'll start off with some introductions and announcements, then we'll move over to the presentations and Q&A portion of today's session. We do ask that you stay muted unless you are speaking. During the session, we do encourage everyone to share their insight and perspectives. You may stay muted. However, you can unmute whenever you do have some insight to share, raise your hand, or use the chat function as well. Finally, please note that no protected health information is allowed in either the chat or during today's discussion. Okay, so the topic of discussion today is global to local surveillance and transmission of Chagas disease in the United States. This is session 3 of a 4-part series. We will be offering 1.5 continuing education credits for providers and nurses. At each session, please make sure to fill out the evaluation survey to claim credits, and the link will be shared at the very end. Just a quick reminder for everyone that is new to ECHO. ECHO stands for Extension for Community Healthcare Outcomes and is a model of lifelong learning and guided practice that connects community-based health workers and a multidisciplinary team of subject matter experts to create a community of practice and learning. The heart of ECHO is a case-based learning concept, so we do remind everyone that we're all learners and teachers in this space. To introduce you to our moderator, Deliana Garcia works from the local to the international levels, addressing healthcare access for migrants and other underserved populations. She founded and directs Health Network, an international case management program that keeps migrants in care across jurisdictional borders. Deliana, the floor is yours. I see you speaking, but you haven't unmuted.
See, and so I'm a learner.
Perfect.
Thank you so much. Today's session, as you heard, was the third part of a 4-part series on global to local surveillance and transmission of Chagas disease in the U.S. To remind you that there will be another session on May the 9th, Chagas disease among people living with HIV and other immunocompromised groups. As was already stated, there'll be 1.5 CME. Next slide. Do I run my own? No. Those have already been reviewed for you, as you know, the entire description with case-based learning, data tracking, an opportunity for those with expertise to share with others, really the hub and spoke interaction, but it is peer-to-peer, an opportunity for there to be collaborative learning. Next slide. And, this is the entire background on the continuing education. As you heard, there'll be a link at the end for you to do the evaluation in case you are interested in receiving those. So I don't know if someone else would like to do a description of the Clinical Directors Network, who is even more familiar than I am, but I will say that I have been a friend to CDN for a couple of decades now. And so it's been very exciting to know about this New York City-based practice-based research network group because they've done an enormous amount with ARC Design Center of Excellence. I think that the practice-based research and learning and all of their work that they have done around Safety Net practice-based research network is really been an innovation and a leader in the field. And so they've been dedicated to improving access to care and clinical outcomes for low income and medically underserved communities. Next slide. This is the information about the fact that you're registered for this third session through the Clinical Directors Network. And as I said, the fourth session will be Friday, May the 9th. Next slide. And so I would like to go ahead and introduce our speakers for today. Their faces are in those little 1-by-1 squares, and we will have them live and larger here for you. I have to be, have to give the statement that Dr. Paula Stigler, Granados will not be joining us today. She was not able for health reasons to join us, and we're really sorry not to have her because she is a subject matter expert in Chagas, has been deeply engaged in starting all of the work that's been done on the Chagas network in Texas. Next slide. This is me. And you heard about me. And so thank you so much for giving me this opportunity today. Next slide. Dr. Lidia Gonzalez is from Barcelona, but she's currently in South Carolina. And her work, her research has been focused on understanding the epidemiology of that vector-borne disease, but in the One Health perspective. So it's the full environment, animals, humans together when it comes to human disease. And so Dr. Gual has focused on understanding the multiple transmission drivers for T. cruzi in endemic and non-endemic regions and how different environmental drivers shift the epidemiology of the pathogen. We've heard some of this in regard to Texas, and so it'll be very exciting to hear from you, Dr. Guad, from your work in that complete One Health perspective, because I think it is certainly the place where many of us need to be focused and haven't been. Next slide. And then we will have Dr. Melissa Nolan, who, in addition to being the associate professor in epidemiology and biostatistics at the Arnold School of Public Health, she's also engaged with the CDC Center of Excellence in South Carolina. She's the head of the infectious disease group and health disparities in that area. She's done a great deal of research, and it's been focused on patient-oriented public health approaches. So we would ask that she also bring all of that expertise to bear, since she is currently working on projects that focus on the epidemiological characteristics of Chagas disease in the US, Mexico, and Central America. Dr. Will you be initiating our speaking for today? Or yeah.
Sorry?
Is Dr. Gual is going to be our first speaker for today?
Yes.
Thank you. Dr. Gual, the floor is yours. Thank you.
Thank you. I don't know if my internet connection might be a little slow because sometimes it just cuts out. I'm going to share my slides. Let me see. Okay. Can you see them? Perfect. Thank you for the presentation. As you mentioned, I focus on the One Health research of disease, especially vector-borne diseases, and I'm going to focus on Chagas disease today because that's what the Echo series are about. But I'm very happy to discuss other zoonotic diseases at any point. I will have my contact email at the end of my slides. Anyone can contact me. But my email might change in the future since I will be transitioning to a new position at Clemson University as a professor there, but I will be always available. Just to start a brief, what Chagas disease is and how it is transmitted. It is a parasitic infection caused by the parasite Trypanosoma cruzi, and it is a vector-borne disease that has several life stages, with some life stages developing in the bug, in the triatomine bug, and some life stages developing in the mammals, in humans or other animals. And what we see is that during the bug phase, the parasite has some stages that allow it to become infective for us. The parasite will be transmitted through the feces of the bug, and these will come in contact with the exposed skin or the wound where the bug was biting us to feed and enter our skin. In the bloodstream later, the parasite will swim and will find an organ to hide, and it will develop as an intracellular form that's called a mastigotes, and these reproduce inside of the cells, and those last for several years and end up causing some problems in the organs. Now, because it's a vector-borne disease, there's some environmental and ecological aspects to this disease. Usually when we talk about transmission, we talk about domestic or peri-domestic transmission to humans. because that's the environment where we live in. So the domestic environment is where inside of the houses where the triatamine is going to be living, is going to be reproducing, and it's going to allow for that transmission cycle to continue, with sometimes our animals, our pets, becoming a bridge to bring the infection closer. Then we have what's called the peridomestic cycle. This cycle occurs in close proximity to our houses, but not exactly in our houses. And there's other animals involved. And I want to point out that one of the interesting things about T. cruzi is that it does require the bug, except on the case of marsupials, which they allow for the parasite to do that bug cycle within their scent gland organs. Now, you can also acquire the infection through other means. You can get orally exposed by ingesting either the parasite or the bug infected. You can have congenital transmission from the pregnant mom to their child. And there's also the cases of blood transfusion and organ transplants that can cause people to become infected accidentally. And so when it comes to the clinical presentation, Chagas disease has three main forms. There's the acute form that occurs right after the infection is acquired. It results in some weeks and sometimes is asymptomatic. In this instance, the parasite is circulating in the blood, so a lot of the clinical symptoms are related to that acute infection phase. But then these parasites find a successful place to hide, and it becomes to that intermediate phase where patients don't really show any symptoms. And years later, 10 to 30 years later, when the mastigotes have reproduced successfully in an organ and cause some failure, we can develop the chronic phase. And it occurs in about 40% of the cases, but not in all of them. Now, the tricky part about Chagas disease is that cases vary. The symptomology is sometimes non-specific. And even though we have those characteristic signs that we have always used to describe Chagas disease, we've later seen that it's not occurring in all the cases. So what we've seen is that depending on the genetics of the parasite or how people acquire the infection, the symptoms will vary. And then the chronic signs will also vary depending on which organ is targeted by the parasite. There's several diagnostic tools available for Chagas disease, and those are depending on what phase of the disease we are. During the acute phase, where the parasite is swimming in the bloodstream, there's direct detection of the parasite, either microscopically or molecularly. But when it comes to the chronic phase, it is a little bit more difficult to detect the parasite. So we have to rely on serologic testing, where we're detecting the antibodies that our body has developed against the parasite. And so there's several ways to detect those antibodies. ELISA and immunofluorescence tend to be the most common, but a lot of times, especially in the recent years, what you've seen more is this screening using a rapid test and then confirmation through other means. The diagnostics are a little different depending on the country, and they change because the guidelines are not very specific, although the WHO suggests to use two different antigen-based diagnostic tests in order to confirm an infection. Generally, it's underdiagnosed and underreported. So when I talk about cases, just imagine it's an estimated based on what we think it is, not the actual amount of cases. And then this disease has some treatment. There's two drugs available, nifurtimox and becnidazole, but they're long treatments, and sometimes it is unsure whether they're effective or not. But I'm not going to go as much on the clinical aspect to it and shift more to the epidemiology. When it comes to Chagas disease, it's a global problem because it affects people in a lot of different countries. But when you see where most prevalent cases are, you see that the American continent is the one that shows more. And that's because that's where the vector, the triatamine bug, resides naturally. Now, because of immigration and global travel, there's a lot of other cases that have been showing on the recent years due to chronic development of the disease in Spain, in Europe, in Japan, or in Australia. And so I want to talk a little bit first about the vector and the importance of the vector species, because it's one of the most important aspects for T. cruzi transmission. And so here in the American continent, we have about 143 species of triatamines that can vector T. cruzi, and they are all potentially infected. However, from those, we only care about 5, 10 of those because the reality is that some of those vectors don't even get in contact with humans at all. And so we have come with the different classification of how triatamine species can be distinguished. We have those that are strictly found in the wild, and those are considered sylvatic species, and we don't tend to think about them as a problem for human disease. They start becoming more problematic when they start entering our houses. So we have the intrusive species. These are the ones that mostly live in the wild, but they might enter inside the houses to feed on, and they can bring infection, but they don't colonize, which means they don't lay eggs, and you cannot find immature stages of that bug in the houses. Domiciliary species and domestic species, sometimes those terms are used interchangeably. Those are the bugs that will find a spot in the house and they will stay there, they will reproduce, and you will find houses that are infested with those bugs. They're going to be laying eggs and they're going to have immatures growing. And those are the species that we care most about because those are the ones that maintain transmission for us, for humans. And so just to see the picture as a whole, we have the sylvatic cycles. Those are the ones that are maintained in nature. And then we have the domestic cycles, the ones that are affecting us the most. When it comes to domestic transmission, there's been efforts since the late 90s to control those insects and to eliminate them from the houses. But the peridomestic environment, which is the outside of the house and that in between of the wild and the inside of the house, is a little bit more difficult to control because those insects can come in and out. And one of the things that I want to point out from here that's more important is that when it comes to animal transmission, our domestic animals and the peri-urban animals are going to be the ones that are going to bring the infection closer and might bring the bugs with them closer to us. And so one of the reasons why One Health is a great approach to this disease is because we have that perfect intersection of human and animal interactions that bring the infection closer. And so transmission in animals in the sylvatic environment has been considered more unlikely to be vectorial. And that is because when the vector bites you, it has to defecate near the bite site in order for the parasite to enter the bloodstream. And so it is a very inefficient transmission mechanism because it requires multiple exposures for sufficient detection or for sufficient infection, sorry. And so it has been seen that actually oral transmission might be the most likely occurring in nature and might be the most efficient way to maintain this parasite cycle. And that is because with a single bug, the animal will acquire enough parasitemia to maintain transmission and even amplify that transmission. And so I will bring more about the oral transmission, but to humans, because when we think about Chagas disease and Chagas disease efforts, we always think about stopping that vectorial transmission and not as much about other transmission that might be occurring from infection for exposures that are not necessarily vectorial. This is from a literature review from a few years ago. These are some of the first instances where we started to identify that oral transmission in humans was quite prevalent, especially in Brazil. Some recent studies have shown that actually oral transmission might be occurring more than we were thinking of, but it's still underreported because there's not as much knowledge on the transmission risk from the how do people become infected from eating a bug, or how do really they get orally infected. And so The only thing that we see that makes oral transmission a little bit more unique is that it occurs more as an outbreak, as a foodborne disease. So you see multiple cases at the same time developing the disease and becoming positive for T. cruzi. And so this study in Brazil, I think, is very interesting and shows why oral transmission should be considered a little bit more. So this is from 2001 all the way to 2021. They used they have a reporting system where they put all the cases and the cause of their disease, and they took out all the Chagas disease patients that had acute Chagas disease and classified them based on transmission pathway, and they divided those cases into two periods. The 2006 cutoff was because those were the years where they started promoting more the control efforts against the triatamines in the houses. What they found with this is that bacterial transmission decreased very significantly, as well as the general incidence of Chagas disease, but oral transmission shifted. And it was actually almost 60% of the cases on the second period were acquired orally. And so this raised the question whether the vector control efforts are actually shifting how the epidemiology of the disease is maintained. So we were thinking about Chagas disease as a vector-borne, but are we now having to think about it as a foodborne disease? And the reason why this question starts to become interesting is when you think about the different foods that are the most associated with oral transmission, they have a high, they're the areas where the bugs like to live in nature. So for example, one of the biggest food or juices more associated with Chagas disease in Brazil is the acai juice or the acai paste. And that is because this grows in this big palm-like trees where the bugs like to hide in between the fruits. And those are collected and processed sometimes in big batches. And the bug can be accidentally squeezed in between those fruits and end up in the juice, or perhaps the feces of the triatamines are in the fruit and these are not washed properly, and so people become infected. It's similar with the sugarcane juice. This is another one that has been very associated with oral transmission of Chagas disease in Brazil. And this is another one that's very related to One Health, because the reason why this one has become such an important source of oral transmission is because they are using land within the Amazon rainforest. They're deforesting that area to build sugarcane fields. And so the vectors that were living in those forests now have a change in their landscape and they're shifting where they're living. And so they end up accidentally being processed with the sugarcane. And so that's a source of infection that would not be naturally thought of. And then the cultural practices of sharing meals and maybe having some accidental bug come in contact or other ways of contamination of the food are one of the reasons why this form of Chagas disease occurs in outbreaks instead of just individual cases. Another example on oral transmission and the increase over time is Colombia. So we have been very lucky to have a very good collaboration with our colleagues in Colombia, where we've done several studies on Chagas disease. And we did this literature review because we found that it seemed that there was an increase in number of cases coming with orally acquired Chagas disease. And if you see in the table, you can see that actually there's a lot more that have occurred after 2010 that you could see before. But that could be biased because of the reporting. So maybe we are more aware about the oral transmission now and we're detecting them more. But nonetheless, that was an important thing for us to investigate and try to identify how people are becoming infected and where. And so similarly, there was a lot of sugarcane juice too, but one of the important sources of orally acquired Chagas disease in Colombia is palm wine. For those unfamiliar, this is not actually wine, it's a liquid drink, but it's made out of this tree that is a palm tree that it's cut open right there in the field, and they let it ferment, and then they extract that fermented drink, and then they use it as a alcoholic beverage. And so during the processing of the palm wine, what they do sometimes is that there's a sort of cultural importance to the first person that takes the drink. It's kind of like an honor to be the first person. So a lot of times before the fermentation has began, people will start drinking from there. As you can imagine, the hygienic conditions are a little bit concerning because they don't really treat those plants. They just straight cut them and open them. And so a lot of times, because the triatomins live in those plants, they are defecating on those plants, and therefore the parasite is there. The other source that's important in Colombia is orange juice. And even though there's no evidence that orange trees are a place for triatomins to live, when there's this big collections of oranges, like you see on the image, that allows for some bugs to hide during the day to avoid contact with humans or people. And so if they are defecating in those oranges, generally speaking, people don't clean the orange before cutting it and making juice. So a lot of time it's just cross-contamination, and that causes oral outbreaks as well. The other source that seemed to be important was the marsupial food contamination, and that is because sometimes there's intrusions in the houses from the Delphis marsupials that enter, and they might be wandering around the kitchen. And because they are also vectors and they carry that infective stage of the parasite in their scent glands, if they get scared or if they pass by through the kitchen and they infect that area or contaminate that area, the person that goes after and cooks in there, they might become infected. And then the other one that we saw that was very important was the cultural practice of drinking and taking armadillo blood as a healing practice from some of the indigenous and communities down there. And so armadillos are important hosts for T. cruzi infection, and because they don't treat that blood and they are potentially carrying the parasite in their blood, when they consume it, they become infected. And so in this overall scheme in Colombia, what we found is that this oral transmission can be sort of classified with either cross-contamination or direct ingestion. And one of the sources that became questionable but has been proven to maybe be a potential source of infection is infected mammalian meat. Now, there's not evidence of humans becoming infected by eating undercooked meat infected with T. cruzi, but it's been seen that it can occur in animal models. And so that is a potential miss or overlooked source of orally acquired Chagas disease. But you might wonder, why is it important to consider orally acquired cases? Well, they present with unusual symptoms that are not necessarily the ones that you would think of when you think about a Chagas disease patient. They come with more GI symptoms and some other complications that are not always present in acute Chagas disease cases. Sometimes they even have early cardiac involvement, and that is because the orally ingested parasite produces higher parasitemia, and so it allows for the parasite to find that target organ earlier in time and then developing the symptoms earlier in time. The other important aspect on Chagas disease is that the diagnostic is not as consistent as it would be with the vector acquired cases. And this is an example of a case report that we wrote a few years ago, where we had two kids that were infected. They were independent from each other, and they had very similar presentation with some differences in the symptoms. But what we saw is that the the similarity between both cases was the diagnosis was very challenging, and it was very difficult for the physicians to consider them as Chagas disease patients because the vector was not there, and there was a very unusual presentation of the case. But there's still a lot that we want to learn about orally acquired cases. We know that they cause higher mortality rates, but we don't know how they might behave in pregnant women or how this might impact other populations. We know that it might be a problem because it could be that Chagas disease occurs in areas where there's not as many triatomine vectors with this maintenance through marsupials. And so it is something that I am very interested in and that we're trying to answer because it's an important source of infection and another important way to maintain that transmission of Chagas disease. But what about here? So I want to shift from what we've seen elsewhere to what we see here and what we can think about when we're here in the United States. And so starting with the vector, here in the United States, there's 11 species that have been described, and they cover almost the entire continental region. From those, there's some that are a little bit more important because they're a little bit more prevalent, but in general, any of them could be potential vectors and any of them could be potentially infected. And so just talking a little bit about each of those, we have on one end Triatoma protracta. This is a West Coast triatomine. This one goes extension from California all the way to Texas, and it's pretty common. And the ecology of this vector makes them live close to wood rat and pack rat nests, and that's what they tend to feed on. They are more common in rocky terrains, but they also have that intrusive behavior. So if people are living in areas where there's high number of rodents around, they might be seeing this vector entering their houses every now and then. On the other coast, from Texas all the way to the East Coast, we have the Triatoma sanguisuga. This is the one that has the largest geographic distribution, and it is sometimes peridomestic and lives closer to urban houses than you would see in other US-based triatomines. This one has a little bit more preference for animals that are urban, like opossums or raccoons, and that might be an important thing to consider because those are animals that we tend to see in neighborhoods and in areas. And so if these vectors are around and they're feeding on these animals and they're infected, there's a potential risk of transmission. They can also enter the houses, and they've been seen in houses, and so they are an important species to consider. Then we have on a little bit more small geographic range, Triatoma gerstacheri. This one is a common sylvatic and sometimes peridomestic species, and it's been only described in Texas and New Mexico, and it's also have that rodent preference, but it's been found around dog shelters. And so it's feeding on animals that are close to us, therefore there's a risk for us to get exposed to this vector. And it's also been seen in south houses, so it's an intrusive species. And then the last species in the United States that I want to highlight is Triatoma rubida. This one also goes on more the southwest area from California to Texas. And it also has that preference for rodents, and it has the intrusive species behavior that we've seen with other vectors. And so we have the vector. Are we having cases here in the United States? The answer is sort of. So it's very difficult to identify active acquired cases in the United States because there's not as much thinking about detecting cases here. Most of the time when physicians diagnose Chagas disease patients, they're immigrants that are coming in, but they're not necessarily thinking about individuals that have never left the country to potentially being infected with Trypanosoma cruzi. But there's evidence that people are getting infected. It is just something that is not emphasized enough. And that is because the United States is not considered an endemic country. And so there's no guidelines, there's no notification. It's not a mandatory notifiable disease. And so we're not constantly thinking about it as a potential infection. And so even though there's a few cases, this is not the only problem. And in the sense that people that are exposed to triatamines here seem to develop other types of responses, such as allergic reactions or very severe bug bites, rather than developing Chagas disease. And so there's a lot to uncover on what is happening in the United States that makes human cases a little bit more rare. And we need to put more surveillance efforts in order to be able to identify what is going on. There's not enough information on the parasite ecology here in the United States as it is in other countries in Latin America. And so there needs to be a little bit more awareness among medical and veterinary personnel to identify those cases. And so are we doing surveillance? What do we know? There's some studies, but there's not a ton of them. And what we see from those studies is that there's infection and it is occurring in many states and it is underreported. This is a study that I think is very interesting and shows that because this tested thousands of dogs across the United States that were US working dogs from military and other agencies, and they screened those for Chagas disease, and they found that they had either antibodies and some of them even were infected. There's no explanation on how these are becoming infected. We know that these are trained and raised in US soil, and therefore that means that these would be autochthonous cases, but because there's not enough infrastructure to do this screening for all the animals that are coming here, that are living here, we really don't know what the actual situation is. On a similar note, there's been some studies on wildlife, and we see that these are infected. Some cases, we see infection rates to up to 60%, which is a lot if you think about it, because these are animals that live very close to us and they're maintaining that transmission. And if we know that the vector is here, we know that this transmission is continued. And if we also know that the Delphis virginiana in this case is infected, we can maintain even if there's no vector. And so it is something that I tend to highlight on why One Health is important, is because maybe we're not testing the people, but we know the transmission can occur because the animals are getting infected. And so it is sort of our duty as researchers to try to find the missing pieces and try to identify where and how we can become more at risk to this disease and other diseases. And so these are examples of a couple of recent papers where people have been finding triatamines around their houses, and they're getting exposed in areas that are not actively thinking about Chagas disease as a locally acquired disease. And so it is important that physicians are aware and consider this during their differential diagnosis, because we could be potentially saving people from developing chronic Chagas disease. And so not just because of the vectorial transmission, but we still don't know what the human risk could be when it comes to oral transmission. And I want to put this out there for people to consider. We need to think about what happens in other countries when it comes to cultural practices that make people at higher risk of orally acquired Chagas. And so here I bring this as almost a little bit of a fun thing to think of. There's people that maybe not frequently, but they tend to eat raccoon or opossum because that's what they were raised on, or because there's a cultural predisposition to that. And if we know that opossums or raccoons can be 50-60% of the population infected with T-cruzi. People could get infected by eating or just by manipulating the carcasses of the infected animals. And so I'm almost reaching the end, but I think it's very important to highlight why One Health is a conjoined effort and why we all need to put a little bit in our end to be able to understand how diseases transmit. It's not only about the risk in humans, it's also about the risk of extending the transmission, increasing the areas where the vectors live, or just being able to detect cases earlier in order to be able to treat them. And it is very important that we develop guidelines for all the medical and veterinary personnel that are actively seeing potential cases in order for them to be able to detect them, as well as for us researchers to try to push on getting funding and try to advocate for those patients that need that voice. And so I will end here. These are my 2 email addresses that you can reach on to me if you have any questions, but I'm happy to take them during the Q&A session after. Thank you.
I have one question, Lydia.
Yes.
From Bernardo Moreno, he said, which mammal reservoirs live in sugarcane fields in Brazil?
That is a good question. I assume it's small rodents, but that is because when I think about sugarcane fields, they are tall plants with not as much branches, but just like providing shade. So a lot of times it serves as a place for smaller animals to hide. So rodents or maybe smaller mammals. I do not know about the species though.
Thank you, Dr. Gual. And so, excuse me. Oh, now I'm going to choke. Please excuse me. We're going to go ahead and move on and invite our next speaker to join us, please. Oh, goodness, please excuse me. Dr. Melissa Nolan. We've spoken a little bit about her and her work and her research in infectious disease and health disparities. I'm going to sign off because I'm going to keep choking. So, Dr. Nolan, if you could please join us.
Yep, thank you. Is it okay if I share my screen like this? When I do the presentation mode, it like disappears. Can you guys see?
And sorry to interrupt. I see that we do have one question from Mark Beatty who asked, can he ask a question? So I think we should let him do that.
Yeah, that's great.
Mark?
Okay. Yeah, hi. So I just a question. The I is this the difference between the species, the triatomine species in the US and those further in Latin America, um, their ability to transmit based on the fact that they're eating and their feeding behavior and their defecation? Because I seem to recall something of that it was very important that certain species did both at the same location, and that led to transmission of Chagas, whereas many of the U.S. native species of trianomine don't do that. Can you clarify on that point?
Yes, I don't know about the specific species, but it is true. So what makes an effective vector is whether they will defecate immediately after they're feeding because they're still nearby the biting site, as opposed to the ones that, get off the animal or the person and then they defecate later on. It is true. Now, this is one of the things that I think is important because when you think about oral transmission, if those triatomine bugs end up defecating closer to the food source, then that is the way of transmitting. And so I think there's still a little bit of an unknown aspect on how are people really getting infected in some of those areas, because vectorial transmission does not necessarily always seem the more feasible, even in cases where there's intrusion.
Thank you. So sorry, Melissa.
And sorry, there was another question from Laura Rothfeld. who is saying, I have seen Chagas cardiomyopathy in working dogs, but does the parasite also infect and cause disease in other animals like opossum and raccoon? So we know that that they are present in those two species, but I don't know whether they can cause cardiomyopathy. I don't know whether that's known.
I believe they do, because of other animal model studies where they study the development of cardiomyopathy on animals that are infected. So I would assume that they do. We might not be able to find them because they might have died before you can, like, there's some surveillance bias in here.
Okay, thank you.
And Mark, I will also address your question. I can speak for Gerstackeri at least. There was thought that it does not defecate while feeding, and that we have shown that to be actually false, that it does defecate while feeding. I don't know about other species, in particular where you live, but at least for Gerstackeri, it does. But I have some hypothesis about that we can discuss. So let me share my screen again. And again, I hope it's okay. I'm going to share my screen in this manner, mostly just because, I don't know, my computer is weird when I go into the present mode. Everything's disappearing. So thank you guys for your patience with that. Okay. Okay. So as Lydia mentioned, there's not much surveillance in the United States. And so today I'm going to talk to you guys a little bit about the work around what we've been doing in the United States and the potential for locally acquired transmission. We're going to talk a little bit about congenital transmission, and then we're going to come back to some ways in which we could strengthen our ability to do local detection here in the US. And as mentioned, my name is Melissa Nolan. I'm an associate professor here in the University of South Carolina. I was lucky to get pulled into Chagas disease during my dissertation work, and I've been really fascinated and lucky to be in this group of folks, including with Paula and others that do research here in the United States. We're small, but we're mighty. And so hopefully all the work that I kind of present today will be of interest to y'all. So getting started first, when we think about what type of surveillance there could be, it's really specifically just blood donor surveillance. That's the only real routine one. And I apologize, school is out today, but my son has promised to be very quiet. So starting back in 2007, 2008, it became mandatory that every single blood donor be screened the first time that they provide a sample. And so it's not performed at every single blood donor screening the way that HIV would, but it is the very first time. And so since that has started, there have been almost 2,500 cases detected of Chagas disease in the United States through blood donors. And interestingly, about 30% of those were actually born in the United States. Unfortunately, they don't ask specifically about travel history, but you can surmise from this that there's the potential that there could be some local transmission that's happening. So we did, as I mentioned, almost 10 years ago now, I can't believe it. We had done our very first study working with local blood donor organizations. So most people are familiar with Red Cross, but there's actually a large number of these individual kind of independent blood donor organizations. So in Houston and San Antonio in particular, they have their own organizations. And so then at this point, it had been over 5 years that they had been doing screening. And so the blood donors sent out letters on our behalf to folks that had tested positive. We sent these letters out in English and Spanish, and we wanted to understand where they might have acquired their disease. And so of 34 people that had participated in both of those, we found that over half of them had evidence of being locally acquired, meaning that we could rule out any significant travel history. Interestingly, most of these folks had never even left their hometown and had spent most of their lives just within that one area that they'd grown up in. And then as you can see in the red dots in the bottom left, so this is a location of where the locally acquired cases are from. And you can kind of see there's little circles around San Antonio and Houston. Those are the major highways that incorporate those cities. So you can see that most of these locally acquired cases were in more rural areas. So that was a common theme that we identified, rural residents. And interestingly, not everyone had reported triatamines, and we were not able to find triatamines around their house. So certainly there's that potential they could be interrupting some natural sylvatic cycles, and that's how they came infected. On the right-hand side, what you're looking at is we also wanted to understand not only are people getting potentially acquired infection here in the US, but is it also causing cardiac disease. Maybe that was why the thought that we haven't identified more of these cases. And so we did EKGs and echoes on those folks. And what we found was that about 40% of them did in fact have EKG findings consistent with Chagas cardiac disease. And this is important because if you think about it, this is a healthy blood donor population. And so the fact that we were already finding evidence of these early cardiac abnormalities was impactful. And then most of those that had EKG findings were locally acquired. And sadly, only one of these cases had been offered treatment. And I think I'll talk about this a little bit more in the next slide. So then we expanded that out to California, where we met with two of the major blood donor organizations, the national ones, but then also some local ones as well. And so again, we recruited folks that had both in English and in Spanish that had tested positive through a blood donor organization. And I apologize, this was actually California, New Mexico, and Arizona. What was really interesting is in the Southwestern study, we actually found a lower autochthonous rate. So it was 17% of blood donors that participated had evidence of local acquired infection compared to Texas, where it was about half of them. And interestingly, even some of those that were now residing in California, when we were doing the risk assessment, most of them, or about 40% of them, had evidence of acquiring it in Texas. So we really thought there's something unique about what's going on in Texas. Yes, and then, sorry, sadly, again, we saw a very low amount of treatment in this same kind of patient population. And a lot of them reported going to their doctors with the actual letter from the blood bank saying that they had screened positive, and the majority of their doctors did not do any additional workup. So certainly some important work that we need to do. And I appreciate all of the doctors that are on this call today, because you guys really are at the front lines and you guys clearly are concerned about this. So kudos to you for taking that extra step to learn more about this disease. So as Mark had asked the question, why do we not see more locally acquired Chagas here in the United States? And so I have a hypothesis about this that we're going to talk about that's alternative. And so what you're looking at here is we do see that, again, geospatial variation in locally acquired Chagas. And so the circles are going to be the percent of blood donors that had screened positive. So the red is going to be locally acquired and the black is going to be imported. And so you see over in California and Arizona, we see less locally acquired compared to Texas. And then the star significant identifies a study that we had done in Big Bend National Park, where this idea kind of came to be. We had been invited out by the Big Bend National Park folks back in, 2017 to screen them. They had recently had a dog that had come down with Chagas disease and had clinical symptoms. And so then the local parks folks were really concerned about this because they were also getting regularly bit by triatamine. So we did a health survey, found that 85% of them had a history of bug bites in this area. And about a third of those triatamines were in fact positive, and yet we did not identify any human cases, which is wonderful. But then this kind of led to a serendipitous surprise. When we were looking at some of these triatamines under the microscope, we found these mites attached to them, and this has not been described previously. And then in speaking with a dear friend of mine, Dr. Ed Wozniak with the State Health Department in Texas, he said, I've seen this before. In fact, this is an image that he had sent me. This is from a NIF. He had found entire life stages of these mites on the triatamine host. So then we said, well, let's understand this a little bit more. So we worked with several partners. And so all of the locations you see are here with the stars, and this is going to be Arizona and New Mexico. We collected triatamines from a variety of different places. We've been doing this over about 5 years from multiple different methods. And at this point, we've collected over nearly 700 triatamines across 6 different species. And what we find is that about 20% of these triatamines do have mites attached to them, which is really interesting. But Triatoma rubida in particular was 5 times more likely to have these mites attached to them compared to the other species, as you can see down here. And then Protracta was another one that also had mites on them. And when we talk about what these mites look like, you can see a couple different images here. We have been trying to find a mite insect expert to help us identify them. They're unfortunately too small to really do molecular ID. And there is a paucity of people that do this, but it's thought to be a particular species called Plumifer. But again, there's really only one person that can study these. And so we haven't been able to identify all of them to species. And it is possible there could be different mite species. But what's interesting is that we find the mites attached throughout their body. So it could be the head, the thorax, abdomen, or legs. And then we found anywhere from 1 to 6 mites attached on the same body, although the legs were tended to be more likely where we would identify mites attached. So what does this matter in the context of locally acquired infection? Well, the question is, does this ectoparasite, so having these mites attached to the triatomines, does it lower their vector fitness, which would then lower their ability to transmit infection locally in the United States? And so what we did look at, one, is there was a statistical difference between the percent of triatomines that were T. cruzi positive with and without mites. So what you're looking at is 1% of triatomines with mites, or sorry, 10% had T. cruzi infection versus triatomines that did not have mites attached to them had a higher rate of T. cruzi infection. And then we also found evidence that a difference in engorgement status. So we found the triatomines that had attached mites were more likely to be engorged, which was really interesting. And so we don't quite understand that, but it's possible that maybe they, we don't really know. It's really interesting to us. We'd like to understand that a little more. Oh, sorry. It's possible that maybe they're feeding more indiscriminately, so they're not completing a blood meal and they're going between multiple hosts, which could then lower their fitness. And so future directions on this, there's really only 2 other articles about this out there. And so you see in the top left-hand corner, this is a group, I believe this one was out of Mexico, where they had done some lab-based studies. So some evidence that with their local species that these mites do lower vector fitness. And then on the bottom right-hand side, this is a group out of Argentina that was trying to identify to say that there's specific triatomines that are more likely to have these mites attached to them, which was similar to what we found. And so we don't fully understand why that is, that some triatomine species are better at repelling these mites versus others. My thought, it's probably associated with their environment. So if you have a triatomine that's really good at living in a tree, that doesn't come in contact with other rodent species versus those that are living like within kind of a den or a pack rat nest, maybe they're likely to pick up these mites because they're within that environment. So you are mostly physicians, and so you want to know, more importantly, what is the impact of Chagas disease on the American healthcare system? We did a study a few years ago where we screened non-ischemic cardiomyopathy patients in Houston that were coming in for clinical care. And so this was at Bentop Hospital, which is our indigent care hospital. And so we specifically worked with Latin American born persons who are now living in the United States. And so again, these are kind of your classic epi case definition of who should be tested for Chagas disease. And so to be in this, they had to be foreign born from a Latin country, now residing in the United States, already diagnosed with non-ischemic cardiomyopathy, but then these patients could not have been previously tested for T. cruzi. And so this is, I apologize, maybe a little small. What we found was that 7% of this population was in fact positive. And one of the nice things about this study was by the end of it, we were running out of patients that we could enroll into the study because the doctors, it had already changed their minds and they had started actively screening these patients that were at risk. So that was a really beautiful kind of outcome that we weren't anticipating, but happy to see. But what you can see here with the yellow highlights is that, interestingly, a lot of those that were coming in were from El Salvador. And I know Shiba Maimondi's group has also found this, that there's something unique about El Salvador, maybe the local strains that are circulating there of the parasite that are potentially more likely to cause heart disease. And so that, again, serendipitously, I was able to meet 2 very important and now very dear friends and colleagues in El Salvador at a meeting. We just happened to sit next to each other at a meeting in Mexico. And so this is the head of research at the University of El Salvador, which is their national university, and then also the Ministry of Health. And when we were speaking with them, they were telling us, we know that there's pediatric Chagas disease in our country. For those that aren't familiar, in the late 1990s, early 2000s, PAHO invested a considerable amount of time and effort in eradicating the most important species, Rodnius prolixus. And so then, as of what happened in El Salvador, they were declared eliminated of this species, and they said there's no more Chagas disease, so we only need to be looking in adults. And so this study challenged that dogma. And so what we found, we enrolled kids that were either coming in with fever or kids that were living in homes that were reporting this vector to the local vector control. And so then from this patient population we enrolled over the course of a year, we had 1,200 kids, and we found that 2.4% of them were positive. And so these were kids that were born after those major vector control campaigns. And so these were definitely more recent infections. And what you're looking at here, this is the epi week. So the number of kids enrolled versus the number that were positive. And you can see that we really identified positive kids somewhat throughout the year. And then just looking at their risk factors. So first on the right-hand side, this is a map of the state of Sonsonate where we had tested kids. And so the darker red is an area where we found a higher number of kids positive. And kind of interestingly about that, that's the ideal elevation zone, an area for Triatoma dimidiata. And so we think that is really the main species in this area in particular that could be causing newer infections. And then when we looked at kind of the classic associations, this is unfortunately is a disease of poverty. And so we identified several poverty-related factors associated with the kids that did test positive. But interestingly, I think kind of this one health focus of what Lydia was talking about, we did see that children who lived in a home with a dog were nearly 3 times more likely to be positive. And that's certainly, we know that dogs are an important bridge vector. They go out into the wild, could become infected, and then they're coming back home. And so they're kind of connecting that sylvatic and domestic cycle. So then we really, in kind of further discussion with folks, the question was, were these pediatric cases vector-borne or were they congenital infections, so vertical transmission? We don't fully know what the rate of vertical transmission is, but it's thought to be anywhere from 1 to 10%, depending on the area. And then some locations, such as Bolivia, are thought to have up to 50% or more of an incidence of vertical transmission. And so then we did a study back in 2022, the first pilot study, in collaboration with the Sonsonate Hospital, but then also with, again, the University of El Salvador. We had two real aims. One, we want to understand what the incidence is of Chagas disease among pregnant women coming in for labor and delivery. But then we also wanted to develop a better diagnostic for those that are in the Chagas disease sphere. You know that diagnostics are a huge issue that we have. We have sometimes up to 22% positive predictive value, meaning that of 100 moms that might screen positive on the first test, only 22 would be positive on the next one. And so that makes it very challenging for clinicians. In particular, qPCR has a pretty terrible rate of detecting this chronic insidious parasite, mostly because we don't see a lot of parasitemia, so it's unlikely to have lots of circulating parasites in the blood, those trypomastogotes. They tend to be that amastogote form that's deep down in the tissue. But digital PCR is one way that we think could increase that sensitivity. So going back to the study that we had done, this was done over roughly 6 months in 2022. It was an initial pilot study run by Dr. Katie Lynn from my team. And so we enrolled over 200 women from across Sonsonate and Huachapan. So on the top right, you can see the red is where we had more women that had enrolled into the study, but we did enroll from across this region. And just to kind of discuss a little bit basics of PCR, of digital PCR, if you imagine we're looking for one parasite that could be in this whole blood sample, it's akin to a needle in a haystack, so to speak. But if you took that haystack and you spread it out across the ground, it would be a lot easier to see that needle versus when it's all aggregated and clumped together. And so on the top, what you're looking at is when we have a sample that we put in with digital PCR, you smooth it out over 26,000 individual reactions. And so instead of doing one reaction like you would with a qPCR, you're actually doing 26,000 individual reactions with that same amount. And so the sensitivity is thought to be higher because of that. And just to kind of give you an example, so what you're looking at here is we do these concentration curves. So a high amount on the left-hand side going down to a smaller amount of spiked particles or spiked blood samples. And you can see right here in the middle, this E1. We can detect as few as 12 individual kind of DNA fragments from a blood sample, which is pretty impressive. So for that first aim, we did identify that digital PCR had superior performance. So of the 6 women that were PCR positive identified, when we look at just the mom's blood, so the top right here, the top lighter gray. So of those that were antibody positive, 5 were positive by dPCR, 2 versus qPCR. With cord blood, 4 samples identified by digital PCR, 0 by Q, and then for placental blood, 2 on dPCR, 1. So we were really pleased with these results, and I will eventually publish these. I promise. I will do my best to get that out because we're really excited about those findings. And then when we look at our seroprevalence rate, so overall, we identified 6% of the moms positive. Half of those were by digital PCR, half of them were by serology alone. And then as you can see, number 90 right here was just consistently positive across. And she was one of the, unfortunately, she was a vertical transmission case. So indicating that if you do have a mom that is positive, by PCR, that could be certainly a high risk factor for successful competent transmission. And then when we look at the birth outcomes of maternal infection, so this is mom having infection throughout her pregnancy, we see that they're significantly more likely to require C-section, which could be an important pregnancy complication, especially if you're a rural physician or you're providing care for women in hard to access locations. In particular, though, major impacts on the babies. Unfortunately, babies born to moms with infections were significantly more likely to have a low APGAR scores, both at 1 minute and 5 minutes, more likely to be admitted to the NICU. And then as you can see on the bottom, they had a variety of different health outcomes, including acute respiratory disease requiring respiratory assistance, and then meconium aspiration syndrome, which certainly some of these are also associated with preterm birth. And typically that's where the story would stop. You know, we do a lot of focus now. WHO has really promoted screening women of childbearing age or pregnant women and their babies, but we forget that these mothers can serve as an important gateway to the rest of their family that could also be at risk. And so we were lucky in El Salvador, their first lady created a new law called Birth with Care. And so with that, there's been a lot of support, not only for moms, but trying to use moms as a way to get access to the rest of the family to provide a general health and well-being. And so then anyone that we had identified in this study as positive, the government Ministry of Health then went out and tested their entire family members. So what you're looking at on the right-hand side, those initial hexagons are the numbers are that patient that had screened positive, the mom, and then the circles associated with those are the number of family members that had been tested positive, and then the orange are those that ended up testing positive. So what you're seeing here is that we identified just in a small number of pregnant women that had screened positive. When we tested their families, 3 of them had additional family members who were positive. And so I would certainly encourage you in your own practice, if you do any routine screening of pregnant women and you identify a positive pregnant mom, please then consider testing the rest of the family, because within my own experience, you're highly likely to identify additional cases, not just the baby, but husbands, other children, etc. And so next steps, we actually were lucky to get funded to continue this. So we have an ongoing study. And with this, we really want to better understand the pathogenic mechanisms of vertical transmission. You know, we don't understand why if one mom, you know, you have 2 moms that are both infected during pregnancy, why does one transmit and the other one doesn't? And so we're doing some very basic science, kind of biological studies with that. We're also validating 2 novel diagnostics, including an AI-generated image recognition of the histopathology slides of the placenta. Is there a way then that we could use these placenta samples to kind of prioritize or triage who we should be focusing on diagnostic efforts if you're not able to test the mom during pregnancy. And then we are also expanding to additional patient populations for pathogenic diversity. And then I wanted to kind of bring this, because this is global to local, is the focus of the talk. So I want to bring this back home to the United States. While very rare, there has been an incidence of vertical transmission. I believe there might have been one other after this that had been published, but we do know that there have been a couple cases of vertical transmission in the US, although there was an important study done in Texas at Texas Children's Hospital by Marvin Edwards, who screened thousands of pregnant women and did identify some positive cases, but did not identify vertical transmission. So we don't know what the rate is. It is rare, but it can happen. Yet, at the same time, I think this was a great paper that was published by Sue Montgomery and Dr. Stillwagon at CDC, where they did some calculations and economic calculations and estimations, and did find that it is highly cost savings and effective to screen on a broad manner. So it's strongly encouraged that you screen any pregnant woman that you think could be at risk for Chagas disease. So then something I think is really important, and certainly you guys, this whole program, the ECHO program, kudos to y'all. The goal is, how do we actually create a sustainable screening program? And so you guys are already here listening, which is wonderful. But if this is something that you get really passionate about, how can you create a sustainable program within your local healthcare system? So we did a study here in South Carolina, and I'm going to talk to you a little bit about, I think, what has made this effective. So when we think about South Carolina, it's not a place that we think about for Chagas disease. I think a lot of folks, really think about that southwestern area in Texas. But interestingly, South Carolina actually has a long case report, or a long history of having infections. And as I think Lydia had mentioned earlier, we identified a case within my first year of joining the faculty here. And we even have records of this going back to the mid 1800s. And as you can see on the bottom right-hand side, these are the different ecoregions. That's the green colors. And then the different circles are positive raccoons. And there have also been positive foxes from this area. So starting with this top one, I think kind of going back to surveillance in South Carolina, what was unique about our study? Well, first, even though I think it's interesting to look for locally acquired, it really is true that Latinx foreign-born residents who are now living in your area are at a greater risk. And so I think focusing within that group is important, at least to start with. And certainly a big challenge with that is, how do you get providers to consider Chagas disease? We know, and I'm sure you guys have heard about this, that less than half of providers have ever heard of it, and even fewer are confident in being able to identify or treat. And then something that's unique about South Carolina is that we rank 28th in Latinx population. So there's been several studies on, kind of how do you do these interventions, and high Latinx populations such as California, Miami, for example. But we're seeing an interesting trend with Latinx populations in our country. First, the southeastern US has seen a 26% increase in this population, and half of Latinx populations are now living in historically non-traditional locations, meaning we're starting to see more and more Latinx populations in more rural, non-traditional areas like Ohio or Montana, for example. And so then we're kind of missing the opportunity to identify these cases. And so then, in essence, how do you create a sustainable screening program for a rare pathogen? Because we know that less than 1% of cases in our country have been diagnosed and treated. So the way that we did it is a kind of a three-part piece. First, we did several grand round lectures, very similar to the talks that you guys are doing and are here today. We also then, I wanted to show the doctors that, hey, you do have cases here in your healthcare system. I'm not just going to talk about it, I'm going to show it to you. So we did a screening program, and we worked with several community partners to get out there. So not just identifying them when they were coming into clinic, but also really trying to engage them in their own clinical or their own community settings. But then what I think was really the most successful part of this is we created clinical champions, and it's not just doctors. We went across the provider spectrum. So I talked to the clinic managers, I talked to the nurses, we talked to the medical billing coding staff, so that they knew how, if they, to charge for a patient that was coming in for clinical care. And I think that was really a big success. And then another thing we did was we were able to connect with other Chagas knowledgeable clinicians so that when we did identify cases, they had another physician to talk to who had worked through that treatment workup. And so we ended up stopping at 71 because we identified so many early on. We had gotten a small pilot grant for this so that we could provide free treatment for these patients. So treatment is, as you may know, is on a sliding scale, but it's more than just the cost of the drugs. They have to get routine blood work up to make sure that it's not too toxic for them. They also have to come in, get EKGs, echoes, et cetera. And so that was something we were passionate about, making sure that they were not disproportionately burdened by being a part of the study. But of those 71, we had 6 that screened positive, 4 came back for confirmation testing, and 2 of those were positive. So again, roughly about 50% of people that initially screened positive are actually considered truly positive by having 2 or more tests. But this is the part that I'm really excited about when we look at the sustainability of this. So on what you're looking at here is the number of Chagas tests ordered each week within the healthcare system. That's Prisma. And so when we go back, 4 years prior to our intervention date, we were having less than 1 test a week. But then after our intervention, we've had a significant increase of that, and that has been sustained over the last almost 3 years. And so to me, I think that's a really incredible finding. So not just, again, I think talking to doctors, but you've got to show them that they have the cases, and you have to create those clinical champions across the spectrum. So not just the doctors, you got to get in with all the people that make the healthcare process workflow happen. And that is my last slide. And so with that, I guess I will take questions.
Thank you so
I will
much.
stop sharing my screen.
So there
There are
is,
some.
okay, Dr. Madras, you can maybe go and ask your question.
You want to say hi?
I just wanted to, oh, hello.
Sorry, there's no school today.
Yeah, that's for me.
There. Sure. I'm Basil
Oh.
Medeiros.
I think we are losing.
Very good.
Maybe I'll read just your questions.
Very
So-
nice. I don't have much of a question as a comment. Thank you very much, because I work in Honduras part of the time, and right on the border with El Salvador, and I'm losing. Yeah, just a comment to say thank you very much.
I apologize. It is
I have
breaking up.
seen
Would
the
you?
disease in Honduras, El Salvador. So I'm at TB clinic now. It's very noisy. Yeah. Okay. I don't really have a comment to put in the corner.
Well, thank you for your work along the border. And I do see your comment that you had put in, and I'll just read it out for others. Excellent point of screening pregnant women as a gateway for family testing. It sounds like you guys do lots of screening of pregnant women. And so happy to see that. And then we have another comment from Dr. Mendoza, who is working with the Chagas Control Program in Bolivia, and she does see a need for screening mothers, babies, and families at risk. So thank you guys for the work that you do. Really, you guys are the true heroes that are out there every day, saving lives. And it sounds like, see a comment from Mark, you guys made Chagas reportable in San Diego last April. That's great. There's no doubt that there is the potential for locally acquired infection, I think, in San Diego, although rare. But the fact that you do have a high Latinx population, it's wonderful to see that it's reportable. Bring more attention to it.
Any other questions for our presenters?
Not a question. Aria, but I would like to, Dr. Wadi, your presentation really made me contemplate the different ways we need to be speaking when taking a history, because we certainly can go from the vector perspective, but how many of us have really enjoyed Gauchana or eaten, all the things that you were describing, and never had a conversation about those as a source of ingestion. So it really did bring into sharp relief for me, at least, the very different perspective of needing not to focus so much on that whole transmission aspect, but looking at ways that there might have been ingestion. Thank you. Are there any other questions or comments? Any final points that either of our speakers would like to make?
I just want to thank you guys so much for having us. It's a pleasure to be here. And again, thank you to all those on this call, because Chagas disease is incredibly important to identify and find these cases early on. So thank you for your interest in this. And thank you, Dr. Gual, for presenting along with me.
Thank you. I think there's a question that somebody is saying, Nida is saying, in making a disease reportable, what is the process and requirements for a state like New York, where Latino population is increasing, but no local transmission is reported? I think this is a very good question. I don't know what the legal process is for a disease to be reportable. Do you know, Melissa?
So every state has the list. As you guys might be familiar, there's the CSTE, Council for State and Territorial Epidemiologists, who have a list of nationally notifiable. Chagas is not on that list. And then every state tends to follow those, but then may add one or two kind of local concerns. So it's really engaging with the folks at your state health department who make those decisions.
This is Mark.
Yes,
Just one additional
please.
One additional comment. Yeah, that's so true. But even counties, their local health officers can make a disease reportable within a county. So in the state of California, Chagas is not reportable yet, but with LA County and San Diego County both making it reportable, we're capturing at least a quarter of the state's population, and that may turn this state to decide to make it reportable for the entire state. So it's definitely a process, but just sharing and attending presentations like this gets people informed and could turn change minds about it. There's been some literature on the states that have done this, so I recommend that.
Yes, there's a comment about Sue and I with the San Diego County EIS helped to get the case definition approved with the CSTE. So I think it does become a county and then an individual state level. I know that there was a previous presentation on working with local health departments, and so it bears reviewing and consideration, and then perhaps working with the larger national organization as well. If any other questions or comments? Don't want to truncate it, because this has all been really wonderful information. And there's Masha has put in there that BU has created is a handbook on setting up screening for those interested. And there's a link in the chat that you can click that will take you there. Just to remind you that there'll be an evaluation, and that if you're interested in receiving continuing education credits, you'll need to fill that out. I believe that there is a slide that will have that link on there. I don't know if we can bring that back up, or if it's something that will just go out to everyone who has registered. But also, it's important to ask you for your input. Yes.
So Melissa included the link in the chat, so you can go to the chat and get the link, but also it will be emailed to everybody who attended the session today.
Thank you so much. I think we're done, correct?
Think so?
Raya?
Yep.
Thank you, everyone. Really spectacular. Greatly enjoyed it.
Thank you.
Bye-bye.